A very good morning to all of you and a warm welcome as we celebrate the International Immunology Day today at our institute. This program is being conducted in association with ICMR National Institute of Virology, Mumbai unit, and is supported by Indian Immunology Society and Mumbai Immunology Group. To begin with, I would like to invite our director, Dr. Gitanjali Sachdeva, to felicitate our chief guest, Professor Dr. Priya Ibrahim, Director and Scientist G, ICMR National Institute of Virology. I now request our Director Madam to deliver her welcome address.
Very good morning. On behalf of the National Institute for Research in Reproductive and Child Health and National Institute of Virology, Mumbai unit, I extend a very warm welcome to you all and our very distinguished guests. The event, as Sadhna told you, that is being organized in association with Indian Immunology Society and Mumbai Immunology Group. And the objective is to celebrate the International Immunology Day 2022. And this day is celebrated just to create more awareness about the significance of immunology. And this year's theme is vaccines. And I would say that the vaccines are the ultimate reflection of the translational potential of immunology or immunology-based knowledge. Interestingly, in terms of historical timelines, it was vaccines that came first. Although I would say that vaccines, they're just a part of immunology, but immunology as a scientific discipline, it took off only in the last quarter of the 19th century. Whereas vaccines, the history of vaccine, if you can say, that goes back to as early as 1000 AD. And if you know that Chinese people, they used to inoculate smallpox. And it means at that time also they had this idea of manufacturing immunity. And it was in 1796 when Edward Jenner came. He used cowpox material to get immunity against smallpox. And you must have heard stories of how in England milkmaids they never used to get smallpox because of their previous exposure to cowpox. And almost 90 years later, in 1885, Louis Pasteur and E. Rox, they developed rabies vaccine using spinal cord suspensions of a rabbit that was inoculated with rabies virus. And that vaccine was used for human use. And during late 1920s or 30s, you can say it was kind of golden era for vaccines. Several vaccines against bacterial pathogens such as diphtheria, tetanus, anthrax, cholera, typhoid, tuberculosis, all these vaccines were developed during 20s or 30s. And in the middle of the 20th century, when the methods for viral propagations were refined or developed, and these methods led to the development of various vaccines for polio, measles, mumps, and rubella. And nowadays we talk about cancer vaccines, vaccines which can prevent or can treat different cancers. We have HPV, we have HBV, which can prevent cancers against like cervical cancer, liver cancer. And to prevent cancers, we have now, or to treat cancer, we have live BCG as well as Syphilis T. So these are the immunotherapeutic vaccines. And several trials are also underway for other vaccines, for other cancers. Efforts are also being made to develop anti-fertility vaccines, or have been made with partial success. You know that these vaccines were directed against the placental products. And researchers are now exploring to have vaccines against even allergies or addictions, like now vaccines against nicotine, or you can say cocaine. They are also in developmental stages. So overall, we have a long history and several successful examples of vaccines. Majority of these vaccines, which are directed against infectious pathogens, they have very successfully led to decrease in the mortality and morbidity. You can just imagine how the world demographic would have looked like had these vaccines been not developed or made available. Now coming to the vaccine because of which we are having this meeting in person or physical meeting. Typically, vaccine development takes 10 to 15 years, and probably mumps was the only vaccines that was very quickly made. But that too took almost 4 years. In comparison to that, COVID-19 vaccine was made like with the lightning speed. You know that India's indigenous vaccine, which was developed by BBIL in collaboration with NIV, it took less than 1 year. Although it took less time, but I won't say it was easy. And we are really privileged that today we have with us a great leader, a brilliant scientist, and very inspiring role model who was in charge of this whole process of SARS-CoV-2 virus isolation, characterization, and vaccine trials in animal studies, and probably much more. So welcome Dr. Priya Abraham, Director NLV. And I urge you to welcome her with a big round of applause. I'm sure it was not easy to grapple with some realistic and many unrealistic expectations, hopes, and pressure. So, but you and your team rose to this great challenge. So, thank you, Dr. Priya Abraham, for saving us from this pandemic or many morbidities associated with this pandemic. So today, Dr. Priya Abraham will share some of the scientific insights which she gathered during this process of vaccine development. We are also pleased to have with us Professor Sharad Dalai from Nirma University, who will be joining us online. He'll be talking about malaria vaccine, and it would be really interesting to know what are the challenges associated with the development of malaria vaccine. And before I end, I would like to thank Dr. Shubha Chiplumkar, President, Mumbai Immunology Group, and former Director Actress, for her valuable guidance and support to this event. I also thank Dr. Smita Mahale, our former director, and Dr. Manisha Madkaikar, Director, National Institute of Immunology, for their gracious presence. So with this, I wish you all a very stimulating day. Thank you so much.
Thank you, ma'am. We now move on to an introduction to the activities of Mumbai Immunology Group by Dr. Shubha Chiplumkar, President, Mumbai Immunology Group and former Director at TRIC. Dr. Chiplumkar is unable to join us today and has sent a message that Dr. Taruna Madan will apprise us.
Thank you, Dr. Sadhna. I'm reading out Dr. Chiplumkar's message that she sends me by email. Good morning and welcome to today's seminar jointly organized by NIRCH and Mumbai Immunology Group to celebrate the International Immunology Day. I wish to thank Director NIRCH, Dr. Geetanjali Sachdeva, for her support and encouragement in organizing this program. Dr. Taruna Madan, who is also the Secretary of Mumbai Immunology Group, has chalked out an interesting program in consultation with other members. We are happy to have with us eminent speakers, Professor Dr. Priya Abraham, Director, NIV Pune, and Professor Dr. Sarat Dalai, Director, Institute of Science, Dean, Faculty of Science, Nirma University, Ahmedabad, to share their expertise and research on vaccines, protective shields against pathogens. Every year on 29th April, this day is celebrated to create awareness about immunology and understand the role of the immune system in health and disease. The COVID pandemic is a classical example of how immunity became a buzzword amongst the general population and created awareness on the importance of immunity, especially the vaccines in combating the pandemic. MIG was established in 2010 to advance the knowledge of immunology and related disciplines and facilitate exchange of ideas among scientists, clinicians, students, and teachers. MIG has been conducting several seminars, lecture series, hands-on training workshops for students and teachers in various colleges and institutes of Mumbai and Navi Mumbai. We also conducted an Indo-US flow cytometry workshop that brought together Indian and international expertise to offer hands-on training in technique. MIT has over 300 live members, and it is the support we receive from our members that becomes a driving force for us to carry out more of such activities. I once again wish to thank our esteemed guests, Dr. Priya and Dr. Sarat, for having accepted our invitation, and I'm sure their work will motivate and ignite young minds to learn and explore more about the field of immunology. I hope you all enjoy this intellectual treat. Thank you, Dr. S.V. Chuplunkar, President, MIG. Thank you.
Thank you, Dr. Chiplunkar and Dr. Taruna. Dr. Abraham needs no introduction, owing to the amazing work done by her and Team NIV during the pandemic. For the benefit of youngsters, I would like to formally introduce our chief guest. Introduction. At the forefront of Indian fight against COVID-19, Dr. Abraham Ma'am led Team NIV to many firsts in the country. Reporting of first cases of SARS-CoV-2 virus, first electron micrograph, first 2 whole genome sequences, isolation of virus for the first time, standardization of COVID real-time PCR, first indigenous COVID coverage IgG ELISA, the first indigenous vaccine in partnership with Bharat Biotech International Limited, and analyzed humoral immune responses against SARS-CoV-2 virus variants of concern detected in India, and most recently, over 50 mutations in the Omicron variant. Madam has been the WHO consultant for formulation of protocols for detection and prevention of hepatitis B virus and HIV infections, WHO consultant to Myanmar to formulate hepatitis virus testing guidelines, member of WHO External Committee for Assessment of Sri Lanka's National Plan in HIV, AIDS and STI. She's also the member of National Task Force on HPV, molecular epidemiology, prevalence, genotype distribution, its attribution to cervical intraepithelial neoplasia, and cervical cancer in collaboration with NIH. Dr. Abraham Ma'am also led the ICMR NIH program in collaboration with McGill University, Canada, and Albert Einstein College, New York, for incidence of HPV in HIV seropositives and in head and neck tumors. Formerly as head clinical virology department, Christian Medical College, Vellore, Madam researched on the genetic basis of host immune responses to hepatitis B virus and chronic liver disease. She is one of the 2 scientists who formulated the national syllabus for DM in virology. She has more than 190 research papers in peer-reviewed national and international journals of repute. Madam has been has many salient awards to her credit. Business Excellence and Innovative Best Practices Academia Award 2020, Integrated Health and Wellbeing Council Janani Awards for Medical Research Leadership 2021, the Rotary Vocational Excellence Award, and Business Today Award for Most Powerful Women. With this, I now request Dr. Abraham ma'am to deliver her talk on Covaxin, evaluation in rhesus macaques and immune correlates of protection.
Please stand here.
Dr. Mahali, Dr. Sachdeva, Dr. Matkankar, Dr. Mohanty, esteemed colleagues here in NIRRCH, other staff, and anybody else who's visiting this institute today for this talk on the National Immunology Day. I'm indeed very humbled to have been given this invitation and to have been introduced in a rather flamboyant way. But I will get on with my business, and I'm going to be talking about the India's first indigenous vaccine and its evaluation in rhesus macaques and the immune response that we have noted in humans as well. I think we were in a position, I think before even I go to details, I want to make a disclaimer. Nothing is possible as just the leader being on top. It speaks and it smacks of very good teamwork. So if we've been able to achieve anything, it is on the, very strong, firm foundation of very good teamwork at ICMR NIV. So we were able to embark on our vaccine journey because we, as you had read in the introduction, we had made some inroads into the virus, detecting the virus very early, much earlier than many other countries. We, of course, had ability to view it under the electron microscope. We were successful, finally, virus isolation. It didn't come easy. Many attempts, failed attempts, and finally on March 9th, almost 1.5 months after we first reported the virus, we were successful. And I give credit to Dr. Pragya and her team, Dr. Anita, who worked very hard on, trying and then meeting success. We also published our first whole genome sequences. Because we were able to grow up the virus, we were able to make our first indigenous whole virion-based IgG ELISA. So this was an IgG ELISA against the whole virion. DG himself, our Director General, gave it the name of Kavach ELISA. And then we were able to standardize the in-house microneutralization assay, the plaque reduction neutralization assay, and also make in-house anti-spike RBD ELISAs and nucleocapsid antibody detection ELISAs. I must give a disclaimer here that all these in-house assays were standardized against the NIBC, the National Institute of Biological Standards and Controls that comes out of UK, the NIBC SERA, control SERA, as well as WHO reference panels. Today, none of these in-house assays have any value unless you validate it against international SERA. So we had, as I had said, isolated it in the virus in March. In April, do we have a pointer? Okay, doesn't matter. I think that's the teacher in me. I like to point at the board. Is the pointer here? I'll have to use my. Okay, it just doesn't matter. It's okay. So we had isolated the virus in March. By mid-April, we had By mid-April, we had selected a good strain, a robust strain of the virus. And mid-April also, BBIL approached ICMR as well as with a copy to me, asking us whether we were happy to collaborate with them in the making of an inactivated whole virion vaccine. So by end April, we handed the virus to them. Whole of May, they worked with this. They grew up the virus in their facility and they inactivated it with beta propiolactone. They gave back the vaccine strain to us for us to characterize and check whether the vaccine formulation was truly inactivated. And then in June to August was the period of the preclinical trials. We started with small animals, then hamsters, and then onto the macaques. And then you know about the phase 2, phase 3, and phase 3 trials that stretched over July to November and onwards. So if I were to home in, this is the formulation that we worked with. Other Chinese whole virion inactivated vaccines have also been used quite widely, particularly in China and parts of Eastern Asia. And then you know the other formulations of the vaccine. But we worked with this particular type of vaccine, a whole virion inactivated virus vaccine. Now, what is special about Covaxin? Covaxin has alum, or it's also called algel, aluminum hydroxide. That is there in many vaccines. It's not unique. But what they added was a compound known as imidazoquinoline. It's a TLR7 and TLR8 agonist, and it boosts the cell-mediated immunity. What alum or algel does is a very good stimulator of humoral immunity. What IMDG does is it boosts the Th1 response. So it gave a kind of a, consolidated stimulation of the immune response, both the humoral and the cell-mediated. So studies, these were mainly done in BBIL, but we worked with rats, mice, and rabbits, New Zealand white rabbits, and Vista rats, and then also what species of mice, not the AG2. I'm suddenly blanking out, but anyway, these Balbsi mice. So we worked with all these three species, and we found that when they made 3 formulations of vaccine. One which had just 6 micrograms of the vaccine antigen with alum. The second was 3 micrograms of the vaccine antigen with alum plus IMDG. And the third was double the amount of antigen with alum and IMDG. And if you look at this graph, you can see the robust antibody response building up over time. And if you compare it against the titers of human convalescent serum, it was pretty good, neutralizing antibodies. Likewise, Th1 response also, there was some pumping up. This is the CD3, CD4, and the CD8 response. There was some pumping up of the cell-mediated immune response as well. So we moved to Syrian hamsters, and this was done entirely in NIV. We got our hamsters from actually NIN. So we're very proud to be ICMR. We took our hamsters only from our sister institute. And mind you, all these were sent to us at a time when, we were just transitioning from the lockdown to still very restricted movement in our nation. So we had to actually, across borders, provide all the certificates so they could cross borders and come into Maharashtra from Telangana, because, traffic across borders was really hard at that time. But we did all of that. Every time hamsters came, I would send all the certification. Hamsters arrived. I thought I would start with the hamsters, so then we will understand the macaques better. With hamsters, it was a 3-dose schedule. Day 0, 14, and 35. So 3 doses of vaccination. And on day 50 was the virus challenge. These animals were challenged intranasally with, again, the 3 formulations of the vaccine I described just now. And group 1 was the placebo group that just received phosphate buffered saline. And after challenge, day 3, 7, and 15, animals were sampled for their blood, nasal turbinates, throat swab, and tissue specimens. Of course, a proportion of them had to be necropsied. And finally, we finished up follow-up on the 15th day. So what we found is, if we looked at, when we talk about IgG antibodies that are detected by ELISA, it's often referred to as the binding antibody. So binding antibodies, we started with, when we gave the 3 doses, we found by the 40th dose, even 40th day, even before we gave the virus challenge, antibodies had come up in the vaccinated group. Deep blue is the placebo, everything else is the vaccinated group. And post-challenge also, you could see the titers were pretty good, whereas the placebo was almost rock bottom. Likewise, I think I missed out the neutralizing antibody, I think. Neutralizing antibody titers also were very good. Have I gone? Yeah, I think I missed that slide. When we looked at the virus load in throat swab and nasal wash specimens, again, there was a significantly lower virus load in the throat swabs of all vaccinated groups, and clearance of throat swab was seen in the placebo group only by around the 10th day. When we went to the gross pathological findings, if you look at the inset on your extreme left, inset A, this is the placebo group. And you can see that there is diffuse areas of congestion, and the lung also showed consolidation. Whereas if you look at insets B, C, and D, these are the 3 vaccinated groups, and we could see that the lung looked pretty healthy. Histopathology-wise also, these insets on your left are all the placebo group. On your right are the different vaccinated groups. And you could see that in the placebo group that didn't have the protection of vaccine, there was accumulation of exudate, the yellow arrow, and the black arrow indicates intense inflammatory response with diffuse alveolar damage and cellular infiltration intense, suggesting interstitial pneumonia. But if you look at these insets on the right, by and large, the lung looked, the histology looked almost normal, but for some occasional congestive foci. So the conclusions was, I'm sorry, I missed the neutralizing antibody slide, but it looks pretty much like the binding antibody. Neutralizing antibodies and IgG antibodies were observed not only up to the time of virus challenge, but beyond. There was faster virus clearance in all vaccinated groups. And clearly, the vaccine candidates seemed to prevent Syrian hamsters from developing pneumonia. And something which I did not show you via slide is that there was lower body weight decline in the vaccinated group as compared to the placebo group that lost a lot more body weight. Now we'll go to the preclinical trials, which is what I was asked to focus on. And before I go to these rhesus macaques, I need to also tell you how we caught these macaques. We had to catch wild-caught macaques. This again was lockdown period. And at a time where, you know, you can get the monkeys where when you go to railway stations, melas, near temples, wherever there's human habitation, because, you know, people are munching food and these guys get the spillover. But these guys did not have anything because there were no human beings in any of these settings. Everybody was homebound. Monkeys did not find food, so they had receded into the depth of the forest. So 2 of our scientists trotted off one day saying we'll come back in 2-3 days' time, capturing the monkeys. Of course, we went through all the regulatory approvals of our own animal ethics approval, the CPC-SCA, as well as the conservator of forest approval. They started with Pune, but they landed up closer to the Nagpur and Nanded divisions, forest divisions, because they had to go by road almost near the Telangana border, where they had to get into intensely forested area to capture the monkeys. So what was supposed to have been a 3 or 4 day trip, they returned 21 days later. And every day I'm calling them, and every day Director General is calling me, where are the monkeys? Okay, so this was quite dramatic. And we finally got these primates. I don't like to call them monkeys. It's more respectable to call them primates. So these primates had a 2-dose vaccination schedule on 0 and 14. And on the 28th day, they got their virus challenge. And of course, they also had a post-infection follow-up of day 1, 3, 5, and 7, and necropsy at regular intervals, again sampling the blood, nasal swab, throat swab, rectal swab, also taking bronchoalveolar lavages and other body specimens, urine, stool, every tissue was sampled, and we were also able to X-ray them because they're little larger animals. So if you look at the IgG response, again, it was building up over the immunization period, post-immunization, and if you compare the antibody response against the Kavach ELISA, which is antibody against whole virion antigen, the S1-RBD, and the nucleoprotein. You can see this is the placebo at the rock bottom. And you can see the 3 vaccinated groups having good IgG or binding antibody response, irrespective of the ELISA that we had employed. Neutralizing antibody likewise built up over the duration of vaccination, remained positive right through the follow-up period. And we also went into the exercise of comparing that neutralizing antibody against the strain that was used in the vaccine. This is the strain that is called the original Wuhan strain, followed by 2 other very varied clades. Now, the Wuhan strain belongs to the G clade, and these 2 other clades belong to the O clade. So what we were trying to demonstrate is that the neutralizing antibody response, or what we call functional antibody response, was there not only to the Wuhan strain, but to unrelated strains of the virus. Not moving. The virus load in throat and nasal specimens was also observed. And again, if you see blue is positive, you can see right through up to the day of follow-up, whereas you could see the virus load was crashing with the other immunized. They had reached baseline with the other vaccinated groups. Likewise, with the nasal genomic RNA that we detected in the nasal washes. In the, across other parts of the respiratory tract, be it the nasopharyngeal mucosa, oropharyngeal mucosa, etc., a high proportion of animals did have genomic RNA detectable in the placebo group, but not in the vaccinated group. We even looked at virus load in extrapulmonary organs. With the placebo group, it was detected in the skin, the ileum, colon, gallbladder, stomach, urinary bladder, bladder, and the pancreas. But one particular group, the group 4, one animal had detectable genomic RNA from the ileum and the cecum. That I think was an outlier, because most of the other vaccinated animals had no detectable genomic RNA across any of these other viscera. We looked at the serum cytokine and chemokine profile. In the placebo group, the IL-6 was significantly elevated in the placebo group as compared to the vaccinated group. In the vaccinated group, group 2, that just got virus antigen and alum, they had IL-5, which was significantly higher. And in the vaccinated group, group number 3, that got 3 micrograms of virus antigen, and the alum plus IMDG, IL-8 was significantly higher. But not much inferences could be made, because you know the number of primates in each group were just 5 in the 4 groups. So these were just observations. We looked at the histopathology. Clearly, this is the placebo group, and it's self-explanatory. The lung was extensively involved across all lobes, whereas the vaccinated group, the lung looked really inflated and healthy. When you come to histopathology, this is the placebo group and vaccinated groups. You could see that the yellow arrow suggests intense inflammatory response, the black arrow suggesting exudates, and the exudates here, and the red arrow suggesting again hemorrhages with hyaline membrane formation, etc. When we looked at immunohistochemistry again, we found that we could detect virus antigen in the pneumonocytes as well as the alveolar macrophages. All of these features seen in the placebo as compared to the vaccinated, where the parenchyma looks quite healthy. So conclusions were that the primates put up a very good neutralizing and IgG immune response against different forms of antigen. And also the IgG titer and neutralizing antibody titer showed an increasing trend. And both radiological imaging, organ virus load, the presence of interstitial pneumonia, and the ability to detect virus antigen was possible only in the placebo group as compared to the vaccinated group. And then, of course, BBIL went on to embark on their phase 1, phase 2, phase 3 clinical trials. We helped them with screening their subjects because we provided them with our RT-PCR as well as our screening antibody tests. And even with some of this, a proportion of their PRNT estimations were done back in ICMR NIV, but they also had developed capacity to do PRNTs. So now I come to the era of when we have had, a flux of variants coming. And that in December 2020 started the detection of the first variant of concern, the Alpha variant. Then last year saw the Delta variant wreaking havoc in our country. And with the end of last year, beginning this year, we know we are in the grips of the Omicron variant. So you can see the Omicron variant is literally hogging the show. And this is the graph of what's happening in India. It mirrors what's happening in the rest of the world. India is no different. So we started interrogating the Covaxin's immune response in human vaccinees against these individual variants of concern. Variants of concern either have higher transmissibility or they have high immune escape, and variants of concern typically can also cause a more severe disease. This is where it would qualify to be a variant of concern by WHO. So here is the original vaccine strain. This is the Alpha variant, the first variant of concern, and this is an O clade, again an unrelated clade to either of these. These both belong to the larger B clade. And when we've looked at the median neutralizing antibody titers, this was not statistically significant. What I'm trying to say is that there was no difference in neutralizing antibody titers, no statistical difference in neutralizing antibody titers against the first variant of concern. When we came to the next variants of concern, that from South Africa came out the Beta VOC, variant of concern, and Delta, of course, came out of our own country. When we compared, again, Beta and Delta against the individuals who had natural infection and those who had been vaccinated, we compared the paired match samples in both these categories against the original Wuhan strain. In black dots is the original Wuhan strain. We found that whether individuals had been vaccinated or had natural infection, with the beta strain, there was about a 3.3-fold lower antibody titer, whereas with the delta, it was about 2.7-fold lower, which was significant as compared to the original vaccine or Wuhan strain. This was inadvertently, this was an occurrence that happened in Uttar Pradesh last year, where instead of getting, 2-dose Covishield, individuals got first Covishield and then inadvertently Covaxin. Okay, so it was an inadvertent heterologous prime boost. That means instead of getting 2 doses of the same vaccine, 18 individuals got Covishield followed by Covaxin. So these individuals are sampled for their for their neutralizing antibody using PRNT, and we compared. In pink is those who got homologous or 2 doses of Covaxin. Blue is 2 doses of Covishield, and this is the heterologous prime boost, Covishield followed by Covaxin. And we compared this antibody response again against 3 strains: the original Wuhan strain against the alpha variant, which is the first VOC, the beta and the delta variant. And what we found was, irrespective of which variant we looked at, the heterologous prime boost had the highest median antibody titers. You can see that here. The pinks and the pink and the blue is lower as compared to this gray color. We have been successful in isolating many strains of the virus, all the VOCs, the variants of interest, the variants under monitoring. So, of course, our first was the success in March of 2020. Then we have isolated the alpha, the beta, the delta. These are VOCs, and variants under monitoring are the zeta, kappa, etc. And, of course, more recently, in December of last year, the Omicron. And we're very happy to say we have over 330 different isolates of SARS-CoV-2 stashed away in our BSL-4 facility. So I want to digress here because I think one of the, there was little more focus on the primate response. So I thought I will go back to a very recent publication, which is looking at the neutralizing antibody response against Omicron and other strains in cynomolgus macaques. Okay. So what they did is they used a prime boost strategy, which was varied. The first strategy was to give 2 doses of the mRNA vaccine, and on week 0, week 3, and on the 14th week, give a booster dose. Next was to give 2 doses of the mRNA vaccine and then pair it with the AstraZeneca, which is similar to the Covishield. The third strategy was to give first AstraZeneca and then the mRNA. And the fourth strategy was to give just 2 doses of the AstraZeneca, and the fifth was the placebo. So these were the 5 groups, and this was the immunization schedule. What they found was with neutralizing antibodies, actually this is a publication in Cell, but I believe they have mislabeled the 8 and the 14. Okay, so I didn't edit it because I wanted youngsters to see how sometimes a wonderful piece of work can be, just a little typo in the wrong place. So you can see yourself that this looks more like week 8, right? And this is week 14, and this is week 18, 8. This is week 8, this is week 14, and this is week 18. But I've just retained it from the publication. And essentially, what they found was that the best response, of course, was against the Wuhan strain, and the least for Omicron. You can see the green, okay, across all the groups. Okay, and you can see here they're showing you the different schedules where you had either option 1, option 2, option 3, or option 4. These are the individual options. So irrespective of options, the Omicron gave the weakest neutralizing antibody titers. In terms of actual titers, if you looked at it, against the Wuhan strain, it was 19,000. Whereas this is after the prime boost, whereas for Omicron, it was 2,000. And against specifically the Omicron strain, neutralizing antibodies were much, much lower. It was 1,900, ending up with, with the last strategy, the AstraZeneca. I'll repeat myself. I think I made a mistake here. Wuhan, when you compared against the Wuhan strain, each of the 4 schedules, 5 schedules are shown here. So these are the neutralizing antibody titers against the Wuhan strain using each of the 5 schedules. So starting with the 3 doses with the mRNA is the highest, and it peters off with, lastly, AstraZeneca, 2 doses. But when you compared against the Omicron specific strain, even in the best of situations with 3 doses of mRNA, it was only 1,901, whereas when you went down to the double dose AstraZeneca, it was only 168. So you can see clearly, irrespective of what those values are, there is almost a 9 to 23-fold reduction when it comes to Omicron as far as neutralizing antibodies are concerned. This was RBD-specific antibody detection using ELISAs. And again, the same typos made here. This should be week 8, and this should be week 14. And you can see the titers against the Wuhan-specific antigen and the antibody titers against the Omicron-specific antigen. You can see here the difference in antibody titers is so glaring. A 9 to 17-fold difference against Omicron. But across the board, irrespective of what the vaccination schedule is there, with the prime boost, there is an elevation of antibodies as compared to the placebo, which is there on the baseline. You can see that all showed elevation in antibodies. Even when we looked at the cellular immune response, both CD8 and CD4, you can see this is week 14, week 16. This is correctly labeled. You can see there is a boosting up of the cellular immune response. Little variations based on the immunization schedule, but Of course, with the placebo, this is the baseline, you can see elevation. So what I'm trying to say here broadly with these studies in cynomolgus macaques, there was a broad neutralizing, binding, and cellular immune response, irrespective of the virus antigen used and irrespective of the vaccination schedule used by these studies. I come back to Omicron studies in humans done in other, of course, was published in Cell by another group. I come back to what was done in NIV, and you can see here that at 6 months after following the primary course of vaccination, what we call primary course is the first 2 doses of the vaccine. You know, it was given with the spacing of day 1 followed by day 28, 4-week gap. So after 6 months after that primary course, we looked at the RBD and the nucleocapsid antibody, still detectable. We looked at the NT across the different, the Wuhan strain, the Delta, Beta, and Omicron, detectable antibodies. But again, you can see here, 6 months later, there is a difference in the PRNT values against Omicron versus the original strain, and also differences across the Delta and the Beta. So this is the in descending order, literally, highest against the classical Wuhan strain, followed by Delta, followed by Beta, and last Omicron. When we then gave them, we then got samples from individuals who had been given boosters, and we got their samples, and we looked at whether the antibodies went up. You can see, look at the values up top here. It's 228 and odd, and here it has become 638. So there is definitely a rise in binding antibody titers when you give individuals the 3rd dose or the precautionary dose. Likewise, the neutralizing titers also have come up significantly. And this is put together to show you how after the 2nd dose, 6 months later, and after booster, across the different variants of the virus, you can see that antibody titers have gone up 19-fold with the Wuhan strain, 16-fold with the Delta strain, 14-fold with the Beta strain, and 18-fold with the Omicron strain, though overall Omicron median levels of neutralizing antibody with Omicron are the lowest. We also attempted, of course, our studies are not based on huge numbers of individuals, cell-mediated immune response, but we looked at Omicron-infected individuals who experienced breakthrough infection. What is the definition of breakthrough? Breakthrough is RNA positivity or even antigen positivity after receiving 2 doses of the primary vaccination, 2 weeks after the 2nd dose. So 2 days after getting the 2nd dose is not breakthrough. You have to wait a minimum 2 weeks, and that is the definition of breakthrough. So these were individuals who picked up Omicron infection at least 2 weeks after receiving the 2nd dose. And what we found was that there was cell-mediated, T-cell immune response in these Omicron-infected individuals, irrespective of the recall antigen. So these were individuals, are those who got Omicron infection. And when we look at the interferon gamma secreting T-cell response, when we use the whole virus antigen or the wild type or the Wuhan strain or the Delta type, against these different SARS-CoV-2 antigens, there was at least 1/3 of them had a good response, showing that the vaccine induces broad T-cell immunity, similar to the cynomolgus monkeys response. There was also higher percentage of B cell and B memory cells and higher NK and NKT-like cells, again suggesting that there is a robust adaptive immune response in these individuals. So these were our forays into cell-mediated immune response. And today we know that cell-mediated immune response has a very important role, not only in natural infection, but also in vaccination, following vaccination. So that's the message. I just want to show you what 2 diagrams which look fairly complex, but I'll try to explain this to you. So these are ribbon models where we have, this is the Wuhan strain, the wild type, and this is the mutant Omicron strain. This is the RBD region, this saucer-like structure here, green saucer-like structure, is the RBD region, and the rest is the spike protein. And this is the ACE2 receptor region, okay, which is in orange. So this is where normally the binding happens between the spike RBD and the Omicron, and the ACE2 protein. And this is where, when we interrogated this with the Omicron strain, we looked at the salient mutations, amino acid mutations in the RBD region of the spike protein of Omicron. And we found that these pink circles suggest higher interaction. That means there's a better inter-amino acid interaction between the ACE2 protein and also the RBD region of the spike protein of Omicron. So colored in pink are suggestive of higher interactions, intermolecular interactions. Colored in teal blue is reduced interactions. So in net, to summarize this, is that with the Omicron mutations, there was a net increase in interactions, which could contribute to higher transmissibility, higher infectivity. When we go to this, this is actually the antibody, a select monoclonal antibody ribbon model against, again, the spike RBD, the RBD region of the spike protein of Omicron. This is the wild strain or the Wuhan. Again, with certain amino acid mutations, there more than interactions gained, there were more interactions that were lost. Now, what does that mean? If there's reduced interaction, it suggests higher immune escape, less poorer binding. Higher binding means the virus will attach better to the antibody or to the ACE protein. Lower binding means it is escaping that attachment. And in the context of a monoclonal antibody, the standard monoclonal antibody is not so efficacious with the Omicron strain. So if we compare these VOCs, variants of concern, head to head, and look at the salient amino acid mutations and how it impacts on transmissibility or antibody escape or immune escape, you see Omicron featuring in almost all of them. And if you look net, Omicron provides the capacity for the virus to have higher transmissibility and higher immune escape. Okay, worse situations in both scenarios. And if you compare all the variants of concern, as far as transmissibility is concerned, in ascending order, gamma the least transmissible, beta following, alpha, delta, and Omicron being the most transmissible. Antibody escape, alpha being the least divergent, so there's less antibody escape, whereas Omicron being the other end of the spectrum, maximum antibody escape. So with that, let me wind up. I think I've taken perhaps more time than I should. I just want to acknowledge my colleagues, Varsha, who did all, Dr. Varsha Pudhar, who did all the RT-PCR testing, getting positives, passing it on to Pragya. Her group was the one who standardized the RT-PCR that is used by many, over 60 to 70% of our government institutions. Dr. Pragya Yadav is the person who heads the BSL-4 facility, who isolated the virus. And once she isolates, she hands it over to Dr. Gajanan, who does all the really painstaking PRNT studies, which take ages, so labor intensive. And also, he's done all the binding antibody studies. Dr. Sarah Cherin, who helped with the ribbon model, she heads the bioinformatics division, and that's Sarah Cherin's team. And with regard to Covaxin, I'm not talking about the many other huge inroads we made in the time of this pandemic. But with regard to this vaccine, there's of course Dr. Pragya and Dr. Gajanan, some of Dr. Pragya's team, some of Dr. Gajanan's team here, Dr. Atanu Basu, who helped with the electron micrograph. I must mention the two heroes, Dr. Dilip Patil and Dr. Gokhale, who were the ones who went into the forest to catch the macaques. And when we had to do primate experiments, we had to do bronchoscopy, sample, tissues from deep down in the respiratory tract. So we took the help of surgeon and also pulmonologists to help us with all the bronchoscopy. And we are very, very grateful to them as well. I'm very grateful to the entire team for, pulling weight at a time when we were called to deliver. So with that, thank you for your patient listening. This is our more recent campus in Pashan, the original campus in Pune. And of course, this is the BSL-4 facility where all the preclinical studies were done, where all the virus isolations have been done, and the PRNTs as well. Thank you for your patient listening.
Thank you, Madam, for apprising us about the research challenges and the extensive work done by Team NIV, as well as the challenges faced for development of COVID vaccine to make it India's first successful weapon against a pandemic. I now request our Director, Madam, to present a memento to Dr. Abraham. Dr. Smita Mahale will be presenting a memento to Dr. Priya Abraham. Thank you, ma'am. Dr. Priya Ibrahim will now be joining NIH. Yes, I request all of you to join us for a cup of tea. इसके बाद सर इसके बाद इनका इंट्रो इसको टाइम टी के बाद में
डॉक्टर सर को हेलो
दिखते हैं क्या वो welcome, sir, to the Immunology Day program. So we will be shortly joining after some time for your talk.
Okay. Okay.
Maybe after 15 minutes, how long? 20 minutes?
yes sir maybe around 20 minutes
After 20 minutes.
Okay. Thank you.
Thank you, sir. Okay, so welcome again to the next session with an immunology stalwart, Professor Sarat Talai, Director, Institute of Science and Dean, Faculty of Science, Nirma University, Ahmedabad. Intro slide. Professor Sarat Talai has done his bachelor's in physics from Utkal University, his master's in biotechnology and PhD in immunology from JNU. Professor Dalai has conducted postdoctoral research for 12 years at NIH, Johns Hopkins Medical School, USA. He worked as a scientist for US military malaria vaccine development program at Walter Reed Army Research Institute. He has more than 12 years of research experience in infectious disease immunology. His laboratory at the Institute of Science is working on various aspects: generation and maintenance of memory T cells towards development of effective vaccines with long-term protection, mapping CD8 T cell responses to Plasmodium liver stage infection, developing chimeric biomolecules for novel vaccine adjuvants, treating cancer through immunotherapy. Professor Dalai is a principal investigator of DST, Drugs and Pharmaceutical Research Program grant for developing a national facility for characterization of biotherapeutics, biosimilars worth 50 million Indian rupees. So, I now request Professor Dalai to deliver his talk. He will be delivering his talk virtually. Come.
Yeah, I'm sharing.
Okay.
Yes.
Good morning. Thank you, organizers, for inviting me to share this talk, along with director in IV. It was really very informative and educational for me listening to this COVID talk. Of course, Madam spoke a recent disease, and I'm going to speak on, a lot of initiative has been taken to develop vaccine to contain, combat this, 150 years old disease, malaria. Here I'm going to highlight some of my, our work that we are pursuing here. The title is T-cell based vaccine, hope for malaria elimination. I'm sure now all of you are very interested to learn about cellular immunology, particularly in the context of COVID infection. I just want to make you familiar with the life cycle. All of you know that this is a multi-stage infection. And in fact, most of the time we give explanation why we do not have effective vaccine against malaria, because it's a complex disease. It has three stages of infection, in human, host, as well as in mosquito. I just want to highlight, although we have quite successfully contained malaria, at least using the drugs, but still the number of cases, if you look at, it's quite alarming. And death rate, particularly among children, is quite high in Africa and sub-Saharan areas. And although the incidence has gone down over the years, but if you look at recent increase, particularly in 2020, is largely happened because a lot of our focus has gone to contain COVID-19 pandemic, and we have neglected malaria in the process also. So, but another most important thing also you have to notice that despite having low death rate, and as you can see on these graphs, and this particularly for India, but the drug resistance has really, become very challenging. And in fact, latest resistance against artemisinin warrants that we must go for an effective vaccine. And as you can see, the drug resistance during 1995, and which was quite limited, but now it is quite widespread, up to 1990. And that make a big case for us to think seriously about vaccine development. Since today we are going to talk about T-cell based vaccine, because that is what is something in everybody's mind to take care of intracellular pathogen. Malaria, at least liver stage, it is intracellular pathogen which can be taken care of by CD8 T cells. So we thought we should go back a little bit to our basic immunology. Once the antigen is presented by antigen presenting cells to, let's say, CD4 cells, which should help B cells largely to undergo class switch, and also macrophages to activate them to take care of the intracellular pathogen in the macrophage. CD8 cells will directly involve in killing these target cells. Of course, NK cells also be part of it, but mainly through MSC1 and peptide complex interaction. CD8 T cells will take care of the targeted cells. This is the similar slide. It's a little more explicit, as you can see that when T helper cells get differentiated, undifferentiated T helper cells differentiate to Th1 cells. So mostly it will help the cellular component, for immune system, and particularly interferon gamma produced by Th1 cells will help activate macrophages. Even they also help even CD8 T cells by producing IL-2 to become more cytotoxic in nature, which will participate in cellular immune response. Whereas Th2 type of cells, when they produce cytokines like IL-4, that will help B cells to produce antibody. As we know, the even follicular helper cells, that are critical in ensuring good antibody responses. In fact, better antibody response should be produced only when you have CD4 T cell response generated. So if you look at the vaccines, we have most of the successful vaccines so far, produce antibodies. There are very few which produce T cells, but three challenging diseases now. Corona has, of course, added to it. HIV, malaria, tuberculosis, where we need strong TSL to, control or eliminate these infections. And my lab basically focus on generation of memory T cells. And as you know, memory is very important, and that is, the main goal of vaccine is to generate antigen-specific memory B cells and T cells. And initially they'll be expanded quite a lot. Then will undergo contraction phase, where we'll be left with very few cells. Of course, they could be higher than your starting naive T cells, but then they have to be maintained, and they should be heterogeneous in nature. I will explain later. But the important thing that when we encounter the infection, or we call challenge the same infection, then they must get reactivated and fight the infection efficiently. So that is the whole purpose of vaccinating, keeping us ready to fight infection. Of course, now for Corona, we have to give because, we are encountering pandemic. So, and here it is required, but otherwise in most of the time we take vaccines when we do not encounter infection, with the idea of generating the memory cells so that they can fight infection efficiently. So when it comes to malaria, now, as we know, there are at least 3 stages, and a lot of efforts have been taken to target at different stages. If you look at initial years of efforts, a lot of research has been done to contain this plot stage infection, but we have not been so successful. And now a lot of people, including Neva Kumar, they have been actually trying hard and to some hard success also to make transmission blocking vaccines, so that when mosquito takes up these gametocytes from the infected plot, so they should not be developed. That is the whole idea. But then the liver stage, in the last 30 years, a lot of efforts have been taken just to develop good, effective vaccines so that we can interrupt this infection. The whole idea is that liver stage is a silent stage, from very asymptomatic, and there are not many hepatocytes get infected. If we can effectively generate, vaccines against it, so probably we'll be more successful. Maybe in future we can combine antigens from various stages and immunize the host, to to have effective malaria vaccine. But these efforts to generate liver stage immunity, started in early 70s, late 60s, early 70s, sensory group from New York University. So initially they started using this irradiated sporozoid, initially X-ray irradiated, then gamma irradiated sporozoid. In fact, they tried in Africa, and there are some success there. And what they found, this is the only vaccine, just like we are talking about, whole virus vaccine. Covaxin is the latest one to contain coronavirus. So the same approach was taken here to using gamma irradiation to irradiate the sporozoid, then immunize the host, and particularly this is done through either through mosquito bite or we inject IV, and it was shown that it could induce sterile protection. In fact, I got interested in working on these vaccines after my training at Hopkins when I came to Walter. So I thought this is the only vaccine which actually gives a lot of, it's a hope, and also it's a good platform, experimental system which induces sterile protection. There is no other malaria vaccine which can induce sterile protection. They can provide clinical protection, but not sterile protection like RTSs. And when it was tried in, rodent models, about 6 to 8 months, in human maybe about 10 months, but then it induces both cellular and humoral response. And over the years of research have actually shown that CD8 T cells are critical. These vaccines induce antibody response, CD4 response, NK cells, and CD8 T cells. And particularly, the protection is associated with presence of CD8 T cells in the liver. If you deplete CD8 T cells, then mice lose protection. And that becomes very interesting. So we wanted to actually address this issue a little bit, deeper. And when I came join the Walter Reed lab, I was told we have to give multiple immunization to induce protection. It was a little bit, worrisome to me why we do not have that many vaccines where you have to give so many, immunizations. In human case, they are giving also 6 to 7 times this Plasmodium falciparum sporozoid to induce sterile protection. Nevertheless, my interest was to generate memory T cells and wanted to see how long it could be maintained, and particularly maintain the protection. And this is where I gave 3 immunizations, 75K priming, and 20 and 20. Then afterwards, we will be challenging at different time points to see whether mice are protected, and that I would like to correlate with the type of the T cells we are generating. So the first question you are asking, when these T cells, particularly effector memory cells, should be induced. And in fact, we do not have much idea about antigen specificity. But we know that a lot of varieties of T cells are generated, probably against varieties of antigens. So I used a very little more simplistic approach. As we know, when naive T cells are experience antigen, they will undergo a lot of changes. And then some of the changes has to do with, let's say, CD38, which is an enzyme involved in calcium, influx. Receptors for interleukin 15 or interleukin 7, they also get modulated, particularly for CD8 T cell cytotoxicity also, which we measure in terms of CD107. These are some of these actually markers get modulated. But what we look at these memory type cells based on CD62L change and CD44. Any cells which will experience antigen, CD44 will go up, and CD62L will go down, they get down regulated, so we call effector memory cells. And those who are maintaining, the CD62L, yet they have CD44, we call, central memory, which are basically true or long-lived memory. Based on these markers, we are actually following these cells, over time. Then, but those are phenotypic markers. We also wanted to know which are the, effector molecules like CD107 and interferon gamma, their production. So basically, using this multicolor flow cytometer, we're tracking these cells, trying to establish that the generation of these cells could be linked to this protection. And in fact, based on these markers, as I show, we are, trying to, make a correlation. And then I asked a question, how many doses is really required to induce sterile protection? And most of the studies will measure the protection after 15 days of immunization. I waited, almost 90 days, in this case, 112 days. So because that is the time memory also get established. And as you can see, we need minimum 3 doses of vaccines. As I told before, in human case, the same vaccine requires actually, 5 doses to induce sterile protection. Then we're asking question, why so many doses are required? What is the reason? And when I looked at, these cells, based on their phenotype and effector molecules, I found that we need at least 3 doses here, because this many, let's say in this case, about 1,000 to 1,200 CD8 T cells having this, phenotype and, their ability to produce interferon gamma is required. And probably that is what is the threshold for providing sterile protection. In fact, later on study which we published in Cellular Immunology, we also found doesn't matter how many doses you are giving, as long as you are generating this threshold of CD8 T cells and their presence in the liver is going to ensure the sterile protection. And in fact, this is what also we have been teaching, to our students in the classroom, that one, it takes several years to develop protection. Children are not actually protected in the first, about 2-3 years. In fact, based on the research over the time, we have found it requires at least 3 exposures. By the time children reach the age of 7 or 8, they seem to be a lot more protected, although we do not call it sterile protection, but at least clinically protected. But once that protection is induced, that means probably the threshold of the T cells are generated, or maybe B cell also. So then that has to be maintained, and it has been shown people staying in this endemic area, even after developing protection, if they continue to stay, they can maintain the protection. And in fact, most of us do not believe that there is a induction of liver stage immunity. There is a presence of liver stage immunity among human. So, and in fact, this review paper we wrote in 2015, I'm just giving that argument that as long as you, based on various studies, not only irradiated sporozoite, people have also used chloroquine, which acts on the blood stage if you immunize host or infect the host under the cover of chloroquine, and then we started generating liver stage response. And this is our argument here was that, particularly if you look at in Indian context, our diet is filled with a lot of herbs and spices. We fortified with a lot of spices, and we thought, and as we are comparing the infectivity versus prevalence, our people in India, our infectivity rate is much way low than most of these countries, affected by malaria. We compare China or Brazil or Africa, whatever country. So our percentage of infectivity or malaria infectivity, is 5 to 7 times lower. That actually tells us that maybe a lot of the food that we take in our house or the spices we take, and they are rich in many compounds like allicin in the part of garlic, even ginger, or we have also curcumin. So many of those compounds, maybe they are modulating this parasite in the liver, and that helps in generating CD8 T cell response. And over the years, after we get exposure, probably now we're expanding those T cells, maybe against broader repertoire of these antigens, and that is helping us, generate, protection. So the liver stage immunity combined with blood stage probably helping us to, at the end, particularly, as we reach the adulthood, so that is providing some kind of protection. But to maintain that protection, we also need exposure, mosquito bite, particularly infected mosquito bite. So we are asking that same question: once we generate this, T cell response, can we maintain it? I give this, example, this in a to our students, and we say in Gujarat, most of the young people either want to stay in Ahmedabad or they want to go to America. But then the danger of, going to America and coming back to Ahmedabad is that you are now susceptible to malaria infection. So we pay a price for it, So what it basically means, when you generate this immune response, if you are not getting exposure time to time, when you go to non-endemic areas, then you lose that protection. So in that context, when you take a vaccine, which would help generate, protection in a short time, but if it is not maintained, then it is no good. And then we are asking, answering this question. And now we follow up these mice immunized with the irradiated sporozoite. Then we are giving a challenge, intermediate challenge, and now trying to follow, their pattern of the T cell response. So before challenging, giving a challenge, intermediate challenge, basically after a short time, I wanted to test how long this protection actually maintain when you are giving only 3 days doses of irradiated sporozoite. And as you can see, that this is based on the T cell response, where we are following these T cells based on their phenotype and also their ability to produce interferon gamma, IL-2. As you can see here, after 210 days, this number of cells expressing, KLRG or CD107 is going down. And particularly when you look at the interferon gamma response, it is very poor among the cells which are, present, let's say after 300 days or so. And when we challenge these mice on different time points, as you can see, mice lose protection after 6 or 7 months. So we repeated the same experiment. Now this time what we are doing, so we gave irradiated sporozoite 3 times, and then after 2-3 weeks we are giving infectious sporozoite challenge, just like we are inducing a threshold of T cells to generate sterile protection, and then we are getting exposed again to maintain that protection. So we are giving a challenge here. And then we did the same experiment. Now we have 2 groups where we have given 3 doses of irradiated sporozoite, another group 1 challenge, or infectious sporozoite challenge following the 3 immunization, and we are looking at the T cell response. So as you see here, particularly if you focus on these panels, this is 190 days, and then 266 days, and then when you compare this is the dose where group where we have given only infectious sporozoite challenge versus another group which has received intermediate challenge. We have lot many number of CD8 T cells. Even the time when we are supposed to lose the protection in case of gamma irradiated sporozoite, now they are being maintained. So what this basically here we are trying to tell that infectious sporozoite challenge is probably bringing in certain changes among these T cells were generated following irradiated sporozoite, and they probably are maintained, and both functionally and probably even number-wise also, they could be providing sterile protection. So then before we ask those questions, answer those questions, we wanted to know whether this phenomenon is true for antibody response. In fact, we had collected serum from these mice which have been given irradiated sporozoite only or irradiated sporozoite following intermediate challenge. As you can see, the antibody response is quite stable even up to 400 days, and suggesting that T cells, it is the T cells which are precipitately going down after 6 or 7 months. They are critical in sterile protection. Although antibody is important, but it could be maintained much longer compared to the T cells. So this is here I would like to summarize that CD8 T cells induced by infectious sporozoite vaccine are key to providing long-term protection, and the infectious sporozoite challenge enhances the maintenance of protective CD8 memory T cells. So now we are asking this question. So what are the possible reasons? What are the defects induced among those T cells? Over time, basically the T cells which are generated, in absence of infectious sporozoite challenge. So one question we're asking, are the cells which are generated here, are they dying? That is why we get lesser number of cells, or they fail to get reactivated when we give them challenge. Or even if they are not, they do not have that defect of getting reactivated, do they fail to home to the liver because T cells which are generated, in the draining lymph nodes, particularly long-lived cells, will stay in the lymph node. When we reactivate them, they would, have to go to the liver in this case to fight the infection. If there is any failure in homing, it boils down to the same, issue that we have cells, but they are not actually homing to fight the infection. So these are the three questions we are asking: whether cells generated following, gamma-irradiated sporozoid versus infectious sporozoid challenge, whether there is any difference with respect to their survival, whether they have any defects with respect to reactivation or homing. So we did this experiment, and as you can see, that the cells which are generated following infectious sporozoid challenge after irradiated sporozoid, they seem to have a better survival, based on the BCL2 expression pattern. So they have a better, survival potential. Then we wanted to know what is their reactivation potential. So then we did this experiment in which we took these mice, after giving irradiated sporozoid following the challenge. Then after waiting, 120 days or 230 days, we are giving another challenge, basically to reactivate the cells which are actually tracking in vivo inside the liver or the spleen. And we are using this as a marker. So basically, as you know, these central memory cells, when they get, when they encounter the antigen, they'll be converted to effector memory cells. And we are using this as a marker of the reactivation. What it basically saying that when we give antigenic challenge, so the central memory cells population will go down, and it should be reflected in higher percentage or number of the effector memory cells. So we have two groups here. RAS group basically given irradiated sporozoid, and RIC here we are giving irradiated sporozoid followed by one intermediate challenge. As you can see here, on 120 days, this change is not so significant. In both the cases, whether they are challenged or not challenged, the conversion of effector memory, central memory to effector, which is going down in this case and reflected in higher, they are very much same. But when you look at the T cell reactivation on 230 days, so here the conversion of central memory cells to effector memory cells much slower compared, in the RAS group compared to, these mice which are given irradiated sporozoid followed by a challenge here, you can see. So that gave us clue that the reactivation potential of these, memory T cells in, irradiated sporozoid immunized mice are much lower, and probably that is contributing to, lesser protection or compromise in their ability protection. And then we looked at this effector molecule CD107 for cytotoxicity and interferon gamma. And normally we give this challenge, and then on day 3 of the challenge, we are looking at this, cytotoxicity and interferon gamma. And 120 days, we do not find much difference. Let's say 15% of cells, in case of RAS, are showing their cytotoxicity, and in this case, 17–18%. It's not a very different, statistically not significant. But 230 days, as you can see, on day 3 post challenge, we find only 5% of cells in case of RAS, are showing cytotoxicity upon reactivation, whereas in the group 15% is much more significant. But then another question we are asking here, that of course these cells are getting reactivated within 3 days, because that is what is required in case of mice, that because 72 hours is the development time of the sporozoid to develop into, liver stage merozoites. And within that time, if the cells are getting reactivated, fighting the infection, they can successfully contain the infection in the liver. But if we give opportunity for the cells to, get reactivated, maybe a little longer time, hoping that cells are there, maybe they are, tolerized, but now giving a little longer time, inducing more inflammation, they could be reactivated. So this is the experiment we did. So we allowed 5 days. In fact, we found that in both the groups, very similar percentage of cells are getting reactivated. But then the problem is that parasite has been already developed and come to the blood stage. So here is the question: you have cells, maybe they are surviving, but then their ability to fight the infection is compromised because they take much longer. So I give this example that when there is a war declared, India-Pakistan border, and we have soldiers in Madras, they are coming, because they have been sent home. By the time they receive call and go to the site, probably it is already delayed. So this is the issue we are again asking, that yes, when you are reactivating the cells, which it is taking longer time, the infection has already established. So what we are saying here, that we need CTLs, which are basically cytotoxicity, because they can take care of the infection directly by eliminating the infected cells. We also need interferon gamma, and for long people have been actually talking about what is the role of interferon gamma. Are they directly killing? And in fact, CD4 is also a good source of interferon gamma. So they can activate these macrophages. So the merozoites which are being released, from these hepatocytes, they could be, easily captured by macrophages, and the activated macrophages will kill them. So you need cytotoxic cells, and also you need the same cells which will be producing interferon gamma. So if CD8 T cells are not producing, but CD4 cells, they can maybe also involved. In fact, it has been shown in absence of CD4 cells, the CD8 T cells are not that effective. So you need both. So then we are asking another question, that the cells which we have reactivated, do they have any defect in homing to the site? No, we are using two chemokine receptors here, which will sense the chemokine gradient, CXCR3 and CXR6. CXR3 basically, it can direct cells to any infected site. But CXR3 is specifically, it will target, direct the cells to the liver. And these two chemokine receptors are measuring on these T cells which are generated in the RAS mice or the REEK mice, basically mice which have been given irradiated sporozoite or mice which have been given irradiated sporozoite following a challenge. And as you can see here, there is not much difference, although numbers are slightly higher in case of challenge, but the expression is quite respectable. So this basically tells us that homing potential is not compromised on the cells, but probably it is the reactivation potential of the cells which are basically making this RAS mice susceptible to infection. And based on this, we conclude from this part that infectious sporozoite challenge definitely helping improve the survival potential of liver cell specific memory T cells. And these memory T cells, in the absence of intermediate sporozoite challenge, take longer time to get reactivated. So therefore, timely reactivation of memory T cells is key to ensuring sterile protection. So we had also other thoughts about these, T cells, why, they are not so long-lived, or their ability to fight this disease, their ability to reactivation is compromised, and which is basically ensured by the infectious sporozoite challenge. So I'm not presenting a lot of data from this, but some idea. Actually, in fact, one of my students who worked on this, now he is a postdoc in UCLA. So he came, spontaneously from his side, wanted to do some study. I said, why not you look at this? So what are the changes occurring when you are giving irradiated sporozoite? So we are generating the T cells, and we are giving one infectious sporozoite challenge that brings in a lot of changes. So is it to do with something with innate immune response? So I tell my students in a very simpler way, look at the irradiated sporozoite. It is also considered as a danger or threat. An infectious sporozoite is also a danger or a threat. And now compare these two, the sporozoite, whether they're irradiated or infectious, they're just like a horse. Okay, one is a walking horse, another is a running horse. So when you now passing that road, we saw one side walking horse, another a running horse. how we respond to this horse, which is basically we consider it as a danger. So this is what is our innate immune response or first line of defense. It is our perception to the threat or the danger. Of course, when we have a running horse, we run away, but our immune system, particularly innate immune system, tries to fight it back. So this nature of innate immune response probably is helping generating better memory T cells. Probably that helps them survive longer, or maybe it brings in a lot of changes, in terms of effective function. So we did many experiments. I'm not going to explain it here because of lack of time. We have published this paper in Translational Immunology. But one unique finding we found, we found, of course, there are many innate immune cytokines being induced. But one important finding we had that when we give infectious sporozoid challenge, compared to irradiated sporozoid, mice, so we get good number of CD4 T cells expressing CD40L. And we know that CD4s are important. If we go back to Mike Bevan's work, when you generate memory CD8 T cells in absence of CD4, so they are no longer, functionally intact. In fact, CD4 T cells, CD8 T cells, memory cells also do not survive long. That is one. And we have also shown CD8 alpha disease, which also express CD40. Those CD8 T cells, probably are getting licensed. We call APC licensing that happens in presence of the CD4 cells. So generation of CD4 cells along with CD8 T cells extremely helpful in providing help to CD8 T cells directly or through antigen presenting cells which are being licensed, and that makes actually the CD8 T cells better memory, long-lived memory cells. So and recently a new idea we are actually pursuing. What we were pursuing, because so far we know that CD8 alpha disease are presenting antigen, cross-presenting antigen, and that seems to be a major pathway in viral infections, even in malaria also. But we have some, thoughts, hypotheses. Maybe some B cells could be involved in generating the, memory CD8 T cells. And this is where it comes to our mind. You have also heard the, previous speaker that we have now corona vaccine, DNA vaccine, RNA vaccines, although they are good, there is no doubt about it. But when it comes to using any, vaccine candidate, protein is always, preferred vaccine, and we are trying to find out whether while generating antibody, whether the same antigen bearing CD8 T cell epitope can also be presented to CD8 T cells, so that both the things can be achieved at a time. We have some interesting data. DBT has actually supported this idea we are pursuing now. In fact, we have interesting observation, maybe some other time I'll present. If that succeeds, then it will really take care of a lot of our, vaccine issue, because when you generate antibody, we are not able to generate CD8 T cells, by the same approach. But, maybe we can engage some particular type of B cells, which could be producing antibody at the same time. They could be also helping generate CD8 T cells, using protein vaccines. So I'd like to thank many PhD students, over the years who have contributed to this work, and many MSc dissertation students. Often they would also work with my PhD students. My collaborator, Dr. Ursula Chris from Walter Reed, and Dr. Agam Singh from National Institute of Immunology. And initial years, I didn't have post-cytometer. Actually, my students used to go to the MSU to use their machine, and funding from DST, DBT, and our state funding agency, GSBT and Goods First, and my university. I'd like to end my talk here. Any questions? I'll be happy to take. Thank you.
Thank you, sir, for enhancing our knowledge regarding the role of T cell responses in vaccine development and how it has been successfully modulated to generate long-term protective response against malaria parasite. Thank you.
Stop sharing.
Thank you, sir.
Thank you. If there's any question, I'll be happy to answer.
Are there any questions? No. Okay, thank you, sir, for an excellent talk. Stop sharing. I now hand over the dais to Ms. Hazra and Pooja for conducting the quiz. This is the most exciting part of today's program. It has been christened as Quizintine, the immunology quiz designed by our PhD and postdoctoral scholars. So over to you, Hazra and Pooja. इसे बंद कर दो। नहीं करते इसको ये बंद कर दो हाँ क्या यूट्यूब का रहने दो
Good afternoon, everybody. So we finally come to our most awaited part of the day, which is the quiz. We've named it Quizzing Time. Before starting with the quiz, we'll ask participants to take our chits and we'll allocate you all into teams based on the chits you pick up. Okay? So Ms. Aishwarya, please.
कैसे रखने वाला हूँ
I will I request all the participants of the quiz to please raise their hands so we can take a head count. Eighteen.
और कितने लेंगे? फिर 11 ही है। वो 3 मिनट एक्स्ट्रा है।
वही चीज
रेटिंग देंगे
तो देखिए करके आते हैं
वो लेके सब लगेंगे ना यहाँ पे क्या तो चेहरे मेरे भी तो पीछे रखो देखो दो पीछे पीछे रखो इधर पीछे रख लेते इस साइड मैं 5 बाकी में 4 अनिल अनिल ये चीज दिखेगा
बैटरी तो
आपकी फैसिलिटी क्या है निकाल रहे हो ना ठीक है तो ये राइट ठीक है तो ना चार ही उनको बोल दो ना 4 ही चाहिए बस हाँ वही 5 कर 5 4 और इधर 5 हो गया नहीं 5 को कैसे बुलाएंगे वी विल हैव टू थिंक उसमें को 2 चीज एक्स्ट्रा बनाना अकॉर्डिंगली वी हैव टू सेट 5 वाला जो है ना 5 वाला जिस ग्रुप का नाम है देखो लास्ट सेटिंग कर रहे हैं 5 6 क्या करो ना उनको ना आके 5 है ना दोस्त 5 विल बी कमिंग बड़ी चीज बनाओ फर्स्ट और लास्ट को दो कर सीजी में ठीक है so you can make them sit like that this is और बुलाते हैं they will have to
So the participants can now come, pick the chicks and call themselves into the teams.
Please come one by
Please come one by one.
tell your name and subsequent
Please tell your name.
Weber. Weber John.
Later.
DAUN.
They come from the
Yeah.
executive. Yes. Just after that, check whether you are in the same statement. If you guys also check whether you are. That's be careful. And
Yes.
Anna, can she join?
Yeah. Aishwarya Patel. Team BC.
So that's Aishwarya from Citizen. I'm from Team BC.
Karan from Team BC. Gaurav from Team BC. Gaurav Paradkar from Team BC.
Ravindralinga from Team BC.
Writing to be time. Get a type of some second.
Samir Khan from MMR. Shiva Kadam from MMR.
Pratik Kewaliga from MST.
Sija Rahate from MMR. Sarita Jara from
Draw down from people.
Sagar Abdikran from
Vivek Pillai from Team OPV. Vivek.
Stella from NEC. Over 5, DPP. Shah from Kriti Gordon from Team DPP. Jyoti Varshay from Team DPP.
ज्योति से पहले
Just check your names.
So good afternoon. These are our participants. We have 4 teams: Team BCG, Team MMR, Team OPV, and Team DPP. We'd like to introduce you to our timekeeper, who's Ms. Aishwarya Rao, and our very alert and smart scorekeepers, Mr. Suri Ganguly and Dr. Ram Bahadur Subedi. These are the general quiz rules, and these will be valid for all teams. So the quiz will be total of 4 rounds. For every question that's addressed to a team, only the concerned team members will discuss, and one of the members can answer on behalf of the whole team. The team will be provided with only one attempt to guess the correct answer and give it to the scorekeepers. Other teams or members are not requested to prompt any question to answer. No question will be passed on among the teams. Okay. So moving on, we start with our first round. The first round is game of options. So these are the rules. This will be a simple MCQ based question round, and there will be only one correct answer for it. Each team will get to answer 2 questions. The marking scheme is you get 2 marks for a correct answer and a -1 for a wrong answer. The time limit per question will be 15 seconds. We'll tell you when the time starts, and we'll also indicate the question which is going to the team. Okay, so everybody ready? Yeah. So the first question is for Team BCG. Which type of vaccine preparation administers antigenic epitopes or fragments? These are your options, and the time starts now.
subunit
Okay, so the right answer is subunit vaccine. This is an explanation for other types of vaccines. So viral vector vaccines make use of a harmless viral vector. DNA or RNA vaccines are based as nucleic acid vaccines. And the whole virus vaccine either contain an inactivated pathogen or a weakened pathogen. The next question is for team. Time starts now.
See artificially acquired.
Okay. So the right answer is, of course, artificially acquired active immunity. So natural active immunity develops upon an infection. Artificial active immunity is upon vaccination. Natural passive immunity is through antibodies passed through mother's milk, whereas artificial passive immunity is through the transfusion of antibodies. The next question is for Team OPD. Which of the following is a toxoid vaccine administered at 5 doses up to 5 years of age? These are your options, and the time starts now. Your time is up. Any answer?
Yeah, we will go with DPT.
Okay. So yes, DPT vaccine is the right answer. So what are toxoid vaccines basically? Toxoid vaccines are formulated from the toxin that is produced by the pathogen, which leads to the manifestation of the infection in the host. The other options that were included were actually examples of live attenuated vaccines. The last question for the first round of MCQ is for team DPT. The DASH vaccine is administered soon after birth, often with the BCG vaccine. These are your options and the time starts now. Final answer? Okay. So yes, hepatitis B is the oral polio vaccine to be administered soon after. We now move on to the second round of the game of options. Again, the question starts with team BCG. DASH is a routinely used adjuvant in vaccine preparation to enhance its immunogenicity. These are your options and time starts now.
Aluminum hydroxide.
All right, we'll accept aluminum hydroxide. It's the right answer. The CDC recommends aluminium or salts of aluminium as one of the most routinely used adjuvants in vaccine preparation. The next question is for team MMR. DASH is a type of conjugate vaccine combining a less immunogenic antigen with a high immunogenic protein carrier. These are your options and the time starts now. The time is up. Any final answer?
The MMRs.
No, sorry, that's the wrong answer. We pass on the question to the audience. Anybody in the audience? Shweta? Yes, Haemophilus influenzae type B vaccine is the correct answer. Shweta, please announce your name. Okay, so we now just have a look at this. So the CDC enlists the hepatitis type, Haemophilus type B influenza vaccine as a type of polysaccharide conjugate vaccine. The next question is for T or PV. Vaccination against DASH in infants is not mandatory in India, but offered in areas at high risk of infection. These are your options, and the time starts now.
So we are going to go with C, meningitis.
You're going to go with?
C, meningitis.
Okay. So meningitis is the right answer. And as recommended by the Indian Academy of Pediatrics, the meningococcal vaccine had to be administered from 9 months of age, but in areas with high risk of infection. This is the last question now, and that will for this round game of options, and this will be for team DPT. Produces antibodies to her unborn baby through the placenta. How long does this natural immunity last after birth? These are your options, and the time starts now. The time is up. Any final answers that you'd like to give? Okay. Unfortunately, that is the wrong answer. Anybody in the audience? Anirudh? Please answer.
Option A.
Okay, so option A, which is 3 to 6 months, is the right answer. Okay, So now we talk about type. We try to understand that the passing of antibodies through the placenta is a type of passive immunity, and it only lasts for a few weeks or months, as described by the CDC. So now we end with the first round, which is game of options, and we ask our scorekeepers to reveal the scores. Team BCG 4 points, Team MMR 1, Team OPV 4, and Team DPT 1. Pooja will do the rules and the person. Now coming to the another interesting round, which is a pictorial round, and the name of this round is picture this. This round will feature pictorial schemes based on generation and development of various vaccines. Each team has to identify vaccine type or method shown in the picture. Each team will get the answer one pictorial question, and the time limit is 30 seconds per question. And marking scheme will be like that: 3 marks for correct answer, and 1 marks will be deducted for wrong answer. Any doubt? So let's start. First question is for team BCG. From the picture display on the screen, identify the techniques for what it is used, and your time start now. Yes, it is a correct answer. Hybridoma technology interest Peter Millicent in 1975, for which they received the Nobel Prize in Physiology and Medicine in 1984. The second question, based on vaccine development, is for team MMR. From the picture shown, identify for the development of vaccine is this technique used, and your time start now. Edible vaccine. Yes, right answer. Edible vaccine. Edible vaccine can be produced by incorporating transgene into selected plant cells. This vaccine could be produced in edible plants such as bananas or potatoes, which would then provide the delivery cycles. Require no adjuvant to stimulate the immune response. The third question is for team OPV. Ready? From the picture, identify for which type of vaccine is this mechanism used, and you can start now.
We will say it's an mRNA vaccine.
Yes. Right answer. mRNA vaccine is a type of vaccine that uses a copy of molecules called messenger RNA to produce immune response. For example, Pfizer and Moderna COVID-19 vaccine are mRNA vaccine. Last question for this round is for team DPT. So the question is, from the picture displayed, identify for the development of which type of vaccine is this method used? Yes, right answer. A viral vector vaccine is a vaccine that uses a viral vector to deliver genetic material coding for desired antigen into the recipient host cell. For example, viral vector vaccine for COVID-19 is adenovirus type 26 vaccine or Johnson and Johnson vaccine. Thank you. Score. So, score people will provide the score. After the second round, the score is for team BCG 7, team MMR 4 points, team OTV 7, and team DPT 4 points. So from the name of the round, I'm sure you must have guessed that this is going to be based on the charades. One member from each team will be invited and allowed to take a chit. Now the chit that you take will bear a vaccine name. Please keep this in mind for the person who's explaining, who's enacting and trying to explain, and as well as the team members that are supposed to guess the name of the vaccine that's being enacted. Okay. Now the player has to use hand gestures and body language to enact the vaccine name. Each team will get to answer one question. So this will be just a one round thing. The marking scheme is 2 marks for a right answer and we get no negative marking. The time limit starting from enacting to the entire guessing will be 60 seconds per question. Any doubts? Before you signal each other on the different signs of the vaccines. Okay, so we'll start with team BCG. Team BCG will require one volunteer team member from yours. I request the other team members to not discuss anything, please. Team OPV, Team DPT, and Team MMR. So while Aishwarya is enacting, I request all the other team members, all the other teams and their members to not prompt any answer. Okay? Yes. So Aishwarya, yes. Your time starts now.
Smallpox. Venom, okay. Toxin. Toxoid. Antivenom. Mosquito. Malaria.
So they've guessed it right. It's malaria vaccine. I request the team members who are trying to guess the name of the vaccine, please speak in the mic. Even the audience feels participated. Next, we invite a member from Team MMR. Yeah, that's the correct answer. Okay, so now from Team OPV itself. Yes, your time starts now.
Smallpox vaccine.
Okay. So the answer is?
Smallpox vaccine.
Okay, that's the right answer. We now request a member from Team DPT. So your time starts now.
Extra words of
Okay. So that's the right answer, and that's all from this round. We now invite our scorekeepers. Chicken pox. We now invite our scorekeepers to tell us the status of the scores.
After the third round, the scores are for Team BCG 9 points, Team MMR 6 points, Team OPV 9 points, and Team DPT 6 points.
We now move on to our final round, and we've kept a special name for it. I'm sure everybody will love it. So this round is I'll be there for you, and obviously the member who will be playing the round and the teams will all be there together to give the correct answer. So this round is basically based on a Pictionary type of activity. One participant from each team will randomly pick a chip from the bowl. He or she will sketch the clues to convey the answer to the team members. Each team will get to answer only one question. Now, for the participant who's sketching on the whiteboard, please ensure that there is no use of any alphabets or any numbers. It has to strictly be based on any graphics or shapes or process that you're trying to explain. Okay? It will be related to vaccine. This could be processes or this could be some type of vaccine. So this will be a mixed bag of terms that we will try to explain to your team. We'll just make arrangement for the whiteboard.
Switch light off. Projector बंद करने को बोलिए. अभी लाइट भी ऑन कर, लाइट ऑन करना, लाइट ऑन करना.
Google से हम meditation on the way.
Time limit will be 90 seconds per round. One round.
Substrate.
Cytokine.
We request the participants to please speak in the mic.
Is it like a B cell? antigen recognition, PRS.
Binding site.
Aishwarya, so the time is up for Team BCG. Any final guess that you would like to give us one word?
MHC, major histocompatibility.
So that was antigen presentation by MX.
So the answer was antigen presentation by MSC, and the team was able to name only half of it, maybe MSC. So we'll leave it in the hands of the scorekeepers how much to mark you. The decision of the scorekeepers will be final. We now move on to Team MMR. Please take the chair, Amanda.
वहीं पे है उसी के ऊपर है
Team MMR, your time starts now.
next vaccine combinant vector with
We ask the participant to please draw and not use hand gestures.
answer
Okay, so that's the right answer. That is transduction. We now request a member of Team OTV to please proceed. The time starts now. Yes, that's the right answer. Moving on to the last team, Team DPT. Your time starts now. Booster dose. So this is the right answer. Booster dose. Okay, so until the scorekeepers are adding up the scores, we'll play a little quiz round with our audience as well. So we'll ask a question, obviously not a MCQ, a one-word question. Raises the hand first, will get a chance to answer. Okay, so here's the question. Here's our first question. Which viral vector was used to generate the Russian Sputnik 5 COVID vaccine? I'll repeat the question again. Which viral vector was used to generate the Russian Sputnik 5 COVID vaccine? Any guesses? Yes. Please call out your name and department and the right answer. So the right answer, as Bhavna said, is adenovirus. Okay, next, an easy one for the audience. Which type of vaccine should not be administered during pregnancy? Which type of vaccine should not be administered during pregnancy? Okay, please stand up. Please call out your name, your department, and the right answer. And the right answer is live attenuated vaccine. Okay, another one for the audience. Against how many serotypes of poliovirus does the OPV provide protection? So we have Bhavna again calling out the right answer. The answer is 3 serotypes of poliovirus. Okay, next question. The anti-malarial Mosquirix vaccine approved in 2021 is a dash type of vaccine. I repeat again. The anti-malarial Mosquirix vaccine, which was approved in 2021, is a dash type of vaccine. Huh? No, sorry, that is the wrong answer. Any other guesses?
From the team members.
Okay, we include the team members. Jyoti.
Inactivated vaccine.
Oh, sorry, that's the wrong answer. Any other guess? Yes, from team BCG, I think.
Subunit vaccine.
No, sorry. Any last guesses? Okay, so I'll give you the right answer. The right answer is recombinant protein vaccine. Okay, so another question. Which vaccine component can stimulate the production of antibodies only when it is bound to a large carrier protein? Okay. Name the vaccine component that can stimulate the production of antibodies only when bound to a large carrier protein. Unfortunately, that is the wrong answer. Any other guesses? But she's made it easy for other people, I think. Anybody in the audience? Anybody from the teams? Yes, from team ECG, somebody wants to answer.
Is it collagen?
Sorry, no.
Is that right?
Yes, Haptil is the right answer. So we have a right answer from team MMR. You will soon receive a chocolate for it. Okay, now the final 2 questions are remaining. So the second last question, which monkey-derived cell line is routinely used in vaccine production? Which monkey-derived cell line is routinely used in vaccine preparation? Yeah, I have. Okay. Yes, that's the right answer. So these are green monkey kidney cell lines, officially also called as Vero cell lines. Okay, so yeah, she's done. Please call out your name and the department. From
CPP.
So this is the last question. Okay, last chance to win a chocolate. For which type of cancer has the BCG vaccine become a standard of care? For which type of cancer has the BCG vaccine become a standard of care?
Lung cancer.
No, that's the wrong answer. Sorry. Yes. No. Sorry, that's the wrong answer. Yeah. Yes, please. Yes, that's the right answer. Please call out your name and department. Okay, so bladder cancer is the right answer. And we are done with our audience round. So okay, so in a short while, we announce our winners. The dignitaries are yet to come. The scores have been totaled up. And we have one winner? Yes, okay, so we have got a winner. Okay. Just hold for another 5 minutes, please. Good afternoon, ma'am. We now request our director, ma'am, Dr. Gitanjali Sachdeva, to please come on the dais and hand the prizes to the winners.
Panel
names of winner and runner-up teams. And I request Dr. Gitanjali and Dr. Vainav to give away the awards. Tell me the names of these teams. Winner team is?
Let's start with the runner-up.
Team BCG is a runner-up team.
So I request the team members to come out in play. To receive the prize.
ये थोड़ा क्लस्टरिंग लग रहा है
Clustering.
नहीं it is clustering right
दो को मुझे पता है, श्रेया का नाम पता है।
Second prize वाला देखो
Second prize तो नहीं runner-up, my dear. अच्छा मैंने नाम भी नहीं लिखा.
We didn't even know who's participating. We didn't get the names earlier.
They like them.
We will write or you can write for them.
you right now
What participate nobody wants. Who should get them?
who is a kaftan
Who
कौन
is
है
the, I mean, most honest person?
so it's one for each
Yes, yes.
oh. That's
So we'll write your names on them. Or even you can write.
Thank you.
Okay, now second, first prize goes to OP. Team OP.
So that's our podium. Almost eliminated, right? Congratulations, all of you.
Thank you.
It's okay. कप्तान
नहीं सर सब लोग एक साथ हैं।
नहीं
चलो
They were 5, no?
Team would be
5, 4.
जाने
They
का
were
थी।
5 or 4?
Five.
This thing they were 4. So they are 4.
Yes, ma'am.
You won. Good.
Congratulations.
is the question sheet here the quiz हाँ मुझे देखना so it is this entire
खाली वो पिक्चर नहीं रहेगा जो हमने वैक्सीन मेथड that picture will be there.